Cancer is a cellular growth disorder that occurs when the cells divide uncontrollably. The causes for the abnormal cell cycle differ wildly. However, they typically follow the same common progression. Most cancers begin as benign tumors, meaning that they are not yet cancerous. Typically, they're not even growing, invading tissues, or spreading. But they may, however, become malignant over time. Additional mutations in those benign tumors may occur in the abnormal cells to stop responding to the cell growth inhibitors that control the cell cycle. When this occurs, the tumors become malignant. At this point, they become cancerous. They're able to spread and invade neighboring tissues. At this point, they may also detach and lodge in different places within the body. This is known as metastasis. A lot of times, this is going to result from mutations of genes regulating the cell cycle. Now, development of cancer tends to be pretty gradual and multi-step. It could take years before a cell is obviously cancerous. So there is a very, very slow progression, which is why if cancer is caught early on, it's typically easy to stop and control if it's caught early enough. So the development of cancer is gradual. A mutation in a cell may cause it to become precancerous, but other regulatory processes within the cell body might prevent it from becoming cancerous at all. Although cancers vary greatly, cells that possess the following characteristics are generally recognized as cancerous. So for one, they lack differentiation. Cancer cells are not specialized and they do not contribute to the functioning of a tissue. They may still possess some of the characteristics of the surrounding tissue cells, but they typically look abnormal. Normal cells can enter the cell cycle about 50 times before they're incapable of dividing again. At some point, we don't just keep copying the same copy over and over again. For example, if you had to copy your entire textbook by hand, one letter at a time, we could copy it, but each time you make a copy, the chance that there's going to be a mistake in that copy increase. And if you copy it, you're going to copy the mistake you made last time. And if you make any mistakes this time, both your old mistakes and your new mistakes will end up in your next copy. And if you make any mistakes in that copy, then all of your previous mistakes will be carried out into the next copy. Because of this, our cells typically only enter the cell cycle about 50 times. After that, they're shut down for good. However, cancer cells tend to be immortal. They can enter the cell cycle an indefinite number of times, and in this way, we say that they are immortal because they can continue to replicate and divide as many times as they want. Cancer cells also typically have abnormal nuclei. The nuclei of cancer cells tend to be enlarged and may contain an abnormal number of chromosomes. They could have extra copies of chromosomes that may be present, and of those there may also be duplicated portions of some of the chromosomes present, which will cause gene amplification or extra copies of specific genes. Some chromosomes could also possess deleted genes, so we may not have just extra genes, but we may have completely missing genes. So these are all sort of dangers of having abnormal nuclei. Another danger of cancer cells is they do not undergo apoptosis. Typically speaking, cells with damaged DNA will undergo apoptosis. And if you remember what apoptosis is, it's programmed cell death when a cell is not behaving as it should, or when a cell does not have an appropriate version of the DNA. Even if the cells do not initiate it themselves, the immune system can also recognize abnormal cells, and it can trigger apoptosis, which can normally prevent tumors from developing. A lot of times our immune system is pretty good at recognizing not just foreign invaders, but abnormal behavior within our own cells, and can induce apoptosis sometimes, but a lot of times cancer cells do not respond. They won't respond to the apoptosis signaling molecules. They won't respond to that trigger to shut down. So a lot of times they'll fail to undergo that apoptosis that's triggered by the immune system. When that happens, cancerous cells tend to form tumors. Normal cells will anchor themselves to the substratum or adhere to their neighbors, and they exhibit contact inhibition, meaning that when they are in contact with their neighbor cells on all sides, they stop dividing. They go, oh, it's a little crowded in here. I'm gonna just stop, and we're gonna just hang out for a while, and we're gonna just keep it nice and quiet. Cancer cells have no restraint. They do not exhibit contact inhibition. They're like, oh, it's crowded in here, must be a party. I'm gonna invite all my neighbors and keep replicating. Woohoo! Yeah, cancer cells are a little bit crazy. They don't follow the rules. The abnormal cancer cells will pile on top of one another. They'll row in multiple layers. They'll ultimately form a tumor because they just keep replicating and dividing and replicating and dividing and replicating and dividing. So cancer cells can undergo metastasis. So as I kind of mentioned before, what happens is that original tumor can fragment. If those fragments get in the bloodstream, that tumor cell can travel to virtually anywhere in the body through the bloodstream. Or if it enters the lymph stream, it can travel to anywhere in the body and get lodged and start growing in other places. So this is a huge concern with catching cancer cells early, not just so you can stop them in their current tracks, but you can prevent them from spreading rapidly throughout the rest of the body. These new tumors can appear in completely different organs and in other areas. Cancer cells can also undergo angiogenesis. Now, angiogenesis, this is kind of a bizarre one. Angiogenesis is the formation of new blood vessels. Tumors soon run out of blood and nutrients as they grow because they're just sucking up all the nutrients in the area. They're kind of like a weed. So they have to find new sources of nutrients, and they do this by sending out signaling molecules that will that form new blood vessels to feed and nourish that tumor itself. It gets greedy, and it will actually start and generate its own blood supply. So that way it can keep engorging itself on your body's nutrients. So here's just a little bit of what that looks like. Here we have a primary tumor. It started off as a single cancer cell. But if it can start growing and replicating. So a cancer in situ is the tumor in its place of origin. It's not moving. It hasn't gone anywhere. However, it is possible that a single mutation could cause it to invade surrounding tissues. Once it invades surrounding tissues, then it becomes a serious danger that it could enter the lymph vessels or the blood vessels. It could then travel through those vascular systems and end up in different areas of the body. This is just a really great section, so I'm going to mention this, but we're not going to spend a whole lot of time on oncogenes and tumor suppressor genes. Normal growth and tissue maintenance depends on a balance between signals that promote and inhibit cellular division. And there are two types of genes that can be mutated, and when they are mutated, they can upset the balance of cell cycle recognition. And that would be the oncogenes. Proto-oncogenes code for proteins which promote cell cycle in various ways. If a proto-oncogene is mutated, it can become an oncogene. Basically, an oncogene just turns it on. It says, hey, we need more cells. We're going to just keep replicating and just keep replicating and just keep replicating. Basically, it's telling the cell cycle to go faster and faster. It's flipping the switch off. Tumor suppressor genes code for proteins which actually inhibit the cell cycle and promote apoptosis in various ways. So if a tumor suppressor gene becomes inactive, it may promote cancer development. Oncogenes are like stepping on the gas. Tumor suppressor genes, when they are mutated, it's like ripping the brakes out of your car. If both proto-oncogenes and tumor suppressor genes are abnormally regulated, we can start to see cell cycles just spiral way out of control. So if the tumor suppressor genes become inactive, they are responsible for inhibiting the cell cycle, preventing the cells from dividing uncontrollably. Some tumor suppressor genes actually will promote apoptosis, but a mutation in that could prevent that from happening, could prevent apoptosis from happening. Like I said, the oncogenes are like stepping on the gas, and a deletion or an inactivation of a tumor suppressor gene is like ripping the brakes out of your car. There's just no stopping it at that point. There are other causes of cancer. So we could have mutations in either our oncogenes, in either our proto-oncogenes or our tumor suppressor genes, but it is also possible to have a mutation that is caused by improperly monitored telomeres. So chromosomes normally have a special material at the end called a telomere, and those get shorter with each cellular division. When they get very short, the cell will no longer divide. It's kind of like putting an alarm clock on your chromosome that says, okay, we have replicated 50 times. At this point, we have probably accumulated an unhealthy number of mutations. We're going to just shut down, and we're going to stop this line from replicating any further. If we have a mutation in the telomerase gene, so the telomerase gene is an enzyme that helps maintain the length of the telomeres. Each time we undergo a cellular replication, the telomerase clips a little bit off. We're punching the punch card, and when that punch card is full, we shut down that cell line. If the puncher is not working, if the telomerase enzyme is not punching that card, we will see those cells continue to replicate over and over and over again. They essentially become immortal because there's no mark on there that says, hey, you've already replicated 50 times, you had your turn, you're done. So the telomerase can not only fail to punch it, but they could actually lengthen the telomeres. They could actually put more telomeres on, or attach more telomeres to the end of that chromosome and cause even further replication. So that's sort of it for our discussion. Please share with me if you have any questions or if there's anything you want to talk about in our discussion groups. I have lots of personal stories to share from just family members who have gone through the process of having and then beating cancer and going into remission for it. So I know that this topic can be very personal. If you do not want to share, that is fine. If you would like to share, that is fine. Please just let me know if you have any questions or if there's any part of this that you want to discuss further, and I'll be happy to talk about it.